because that means the homologous chromosomes lying at the equator, which is in construct to the mitosis phase, which is also metaphase, which the homologous chromosome align at the equator in a fashion way, and also the spindle fibers attached to the chromosomes, and also it is always randomly arranged in this stage. Thank you.
This stage, the this the spindle fiber attached to each chromosome of each chromosome pair is now separated oppositely.
Telophase is the final stage of cell division. The separated chromosomes, which were put apart during anaphase, reach opposite ends of the cell, forming two distinct nuclei. A nuclear membrane then reforms around each set of chromosomes, and the chromosomes then slowly change back into chromatics. Cytokinesis occurs with cytokinesis, which is division of cytoplasm into two daughter cells. Thank you.
I'm here to explain the result of meiosis 1. At the end result of meiosis 1, we're going to have the number of chromosomes. Imagine chromosomes like pairs of shoes, one from your dad and one from your mom. Meiosis takes a cell and divides it into meiosis takes a cell each with only one set of chromosomes. Meiosis takes two cells each with one set of chromosomes, which are called the diploid, and divides it into two cells with one set of chromosomes, which are the haploids. And the end result of meiosis, we have the mixing up of genes. During meiosis 1, some pieces of DNA mixes with each other. This process is called crossing over, which is why sometimes siblings look alike. Number three, we have the two brand new haploid cells. So the two new cells occur when the diploid cells have divided into two. And the importance of meiosis, it is essential for sexual reproduction, where the sperm and egg cells join together. So that the baby will have the right amount of chromosomes. And it is important for genetic diversity, so as the organism will be able to adapt to different environments.
Moving on, we have prophase 2. Here in prophase 2, the nuclear envelope breaks down and disappears. Then the chromosomes condense further. Then after that, the centrioles are formed. After the centrioles are formed, then the spindle apparatus is used to separate the sister chromatids. Then each chromosome is pulled towards the metaphase plate. This stage ensures that each chromosome contains the essential number of gametes essential for species survival.
As we all know, meiosis can be divided into two stages, which is meiosis 1 and meiosis 2. And while under meiosis 2, I want to talk about the second division, which is metaphase 2. In metaphase 2, as you can see here, the nuclear membrane gets dissolved into the cytoplasm. The nuclear membrane gets dissolved into the cytoplasm, which makes it less visible. And which you can also see in the second diagram here, the nuclear membrane is not to be found because it has been dissolved into cytoplasm. Then the chromatids become more distinct, which means they become visible. As the chromosome they get pulled to the center of the cell by the chromatids by the centromere.
So anaphase 2 is a phase within meiosis 2 where the sister chromatids separate at the centromere and are pulled apart by spindle fibers. There are key points to note in anaphase 2, which are first key point that occurs in meiosis 2. The main purpose of anaphase 2 is to separate the sister chromatids. importance of importance of anaphase 2 is encourages genetic diversity. Thank you.
Telophase 2 is the last stage of meiosis 2 following anaphase 2. It results in they result in the four daughter haploid cells. The chromosomes envelope reforms around each set of the cell. Then the cytokinesis occurs. In the cytokinesis stage, the cell